Hi, today I'm going to present a very interesting topic, which is metatranscriptomics. So basically, I'm going to present introductory idea, pipeline, and the details of metatranscriptomics. So metatranscriptomics is the study of RNA transcripts obtained from environmental samples, which provides insights into the functional activities of microbial communities. If you want to know the details, introduction, or more definition, then you could understand metatranscriptomics is the study of total RNA, that is transcriptomes, in a microbial community, which offers insights, deep insights into which genes are actively being expressed and how the microbial communities are functioning with the respective environment. It also allows researchers to understand how microbial populations respond to environmental changes, identify active metabolic pathways, and assess the overall health and activity of a microbiome. So, metatranscriptomics is the characterization of RNA content for the selected samples, as opposed to the DNA content, which is analyzed using metagenomics approaches. Similar to metagenomics approach, metatranscriptomics could be used to identify microbial members of an ecosystem while simultaneously obtaining functional insights about a microbial community. Because metatranscriptomics utilizes RNA transcripts rather than DNA, it can provide information about the dynamic nature of a community and allows conclusions to be drawn about how changes in the surrounding environment induce community-wide alterations at the gene expression level. So if you are interested to know the details, so this basically definition gives you the differences between metagenomics as well as metatranscriptomics. If you will listen my previous videos from metagenomics, then you could easily understand in metagenomics, you try to identify the microbes based on the sequences, DNA sequences. While in metatranscriptomes, you are studying the total RNA for the environmental samples. Here you could go for gene expression analysis, similar to the eukaryotic organisms. So, for example, functional notation, gene co-expression network, gene expression network, many more things you could do by using metatranscriptomics studies. Now, coming to the next part, if someone is interested which papers to read and easily understand, then these are the three review research papers. You could go and download and read these papers. This is very easy to understand. I have not included any complicated papers. So these papers could be easily understood for meta transcriptomics purpose. And it will help you a lot to immediately start your project or project work related to meta-transcriptomics studies. So please listen both the videos carefully, my previous videos on metagenomics and these videos on meta-transcriptomics, which will help you to study the environmental samples at different levels. Now, the importance of metatranscriptomics, it could reveal active metabolic pathways and helps to understand microbial responses uh, due to environmental changes, complements metagenomics by focusing on gene expression patterns or gene expression profiling. Now, challenges. First thing, RNA instability, then host RNA contamination, bioinformatics or computational pipeline complexities. It requires high sequencing depth. So it could be slightly cost effective. Now, Metatranscriptomics workflow in terms of just wordings, if you want to know without any software's knowledge, then just in summary, first step sample collection, RNA extraction or RNA depletion, cDNA synthesis, sequencing, and finally, bioinformatics pipeline implementation for data analysis. Now, sample preparation and RNA extraction, that is RNA stabilizer or RNA ladder. Then DNA removal with DNase, quality control by using bioanalyzer or nanodrop. Next part is library preparation and sequencing, RNA depletion, rRNA depletion, strand-specific rRNA, the RNA sequencing, platforms, which platform you want to select for sequencing purpose, Illumina, Nanopore, or PacBio. Now, bioinformatics pipeline, that is quality check, Thermomatic by using the software Thermomatic. Then assembly, for which you can use Trinity or MegaHit. Then mapping by using Botai or HiSat2, quantification by using feature count, Salmon, then annotation by using eggnog or Keck databases. Next part is in functional annotation, go for gene annotation, go for Keck pathways, go for domain, protein domain classification, PFAM domains classification or understanding, then homology by using Diamond or BLAST software. And finally, if you want to understand the overall pipeline, how to process, then here there are some of the softwares named which you will require, and it is easy to execute and learn how to run the pipeline for meta transcriptomics purpose. So first thing is process, collect the raw samples or raw files, then go for quality assessment by using fast QC software, then go for generating a de novo sequence assembly for which you can use Trinity software. And by the way, please remember minimum 32 GB RAM will be required for running de novo sequencing. So for 16 GB RAM, it will be quite difficult to manage unless otherwise you have some dedicated graphics card in your computer. And if you don't have graphics card in your computer, then just be prepared that your system has 32 GB RAM. Then go for, after running Trinity, go for transcript expression quantification. And there, here you have homology approach also, for which you can use BLAST homology. Then go for transcript expression quantification, where you can go for abundance estimation by using Callisto software. So this is the software name Callisto. Now construct an expression matrix, and after constructing the expression matrix, go for expression analysis and functional annotation, and for which you have large number of softwares available. So now tools in general for used for meta-transcriptomics purpose, RNA extraction kits, RNA depletion kit, sequencing platform, then bioinformatics tools, for example, FastQC, Thermomatic, HiSat2, feature counts. If you want to know more details, you can write the comments in the videos. We can immediately help you out and guide you. Then go for data analysis for the metatranscriptomics data, quality control for raw reads, then read alignments to the reference genomes, then transcript quantification, differential expression analysis, functional notation. By the way, please remember, here mostly we go for, we perform de novo assembly, but of course, if we have reference genome available, then we can also go for such things, but which is quite rare. So next part is application of metatranscriptomics. It is used for environmental microbiology, human microbiome studies, agriculture and soil health, bioremediation, then disease diagnostics and therapeutics purpose. Next, tips. What are the tips and best practices? Prevent RNA degradation, efficient RNA removal, combined with metagenomics for better insights. Useful resources, MGRAST, EMBL, EBI, IMGM, tools, human, N3, Metatrans, SAMSA2, NCBISRA, Galaxy. These are the tutorials. Then there are some more things which have not been mentioned here, and they are quite common. That is UniProt, which you mostly need it. So be ready that you are aware of these small tools. Now, sample figures for meta transcriptomics. So whatever I have explained in the previous slides, there are, I have presented the professional figures which have been either published by our collaborators or are under publication